going to start. So the title of the presentation, I put like considerations for researchers in the development of CE-IVD assays. So the idea is to provide you with some more solid background information, basically what gap researchers have to take from an assay that they have developed in their laboratory to take this into an actual routine diagnostic IVD test. The focus will be the regulatory framework that we have here in Europe, the so-called IVD regulation. And based on that, yeah, I will give you a few more insights. No. So I just start very easy and simple. So what is an in vitro diagnostic medical device? All of you know, you can take samples from in this case persons, means humans, and then you undergo a certain analytical process, which includes extraction of the analyte, determination, can be a qualitative or quantitative determination, and then the interpretation of the data or result. And then the result needs to be passed on to either medical staff or the patient directly. So in the IVD regulation, this device comprises things like reagents, reagent products, calibrators, control materials, kits, instruments, also software that is intended to be used in vitro. That's why IVDs, for the examination of specimens derived from human bodies. So it's clear the IVD regulation is only applicable to human diagnostics, not to veterinary diagnostics or anything else. And the purpose of this testing is to provide information, as the standard says, things like physiological processes or states, some congenital impairments, predisposition to certain diseases, also safety and compatibility to the potential recipients, if you think about blood donations or organ transplantations. And so on and so on, including also defining monitoring therapeutic measures. And of course, among them also testing for infectious diseases. But this is really, if you think about the whole scope of IVD devices, is just one of many other aspects of IVD tests. And also, it's explicitly said that specimen receptacles are also deemed IVD devices, although they don't generate any results in itself. So with that background, I just want to give you a little more information about this regulatory framework that we have here in the European Union. It's since 2017, the EU regulation 2017-746, that's the official naming or numbering, got into force. It's an EU law binding to all member states, and it took over the previous directive, the IVD directive, which was just a directive that had to be translated or transposed into member states' law. And there are still some transition periods from legacy devices that have been on the market under the old framework, and they can still remain up to 2029. There are certain staged transition periods for different risk classes. These old devices can still be on the market. So what are the most important changes? They are important to understand because then you will see that from them you can derive a lot of requirements that need to be considered for actual CE-IVD test development. So the most important one is the device classification has completely changed. It was a fixed list-based system. There was a couple of analytes like HIV, HBC, HCV, CT, so chlamydia, and others, very fixed. And these are considered or have been considered high-risk devices. And anything else was just, not a listed device. And so it was kind of self-certified. So no one except the manufacturer had to look into the quality of these devices. So that has completely changed. Now it's a risk-based classification. I will show you in the next slide what this means. And then also due to this risk-based classification, the number of devices that has to undergo an external certification process has massively increased. People say it used to be like 10% of all IVDs had to undergo external certification, and now it's like 90%. So it completely changed to the opposite. Also, the IVDR is defining the market follow-up measures to be conducted by the manufacturer much more or much better and more comprehensively. And also lab-developed tests, also called in-house tests, are much stronger regulated. They are not forbidden, as some people think, but they are still allowed, yet much, much stronger regulated. But this is not a topic of today's talk. So here you see an overview of the risk classes. Let me try to get the laser pointer. So we start, or the IVDR foresees 4 different risk classes, starting with the lowest risk, which is A, and then increasing B, C, and D. So the highest risk class means, or the characteristics of it are, it's a parameter that comprises or displays a high individual and a high public risk of infection, of disease, and so on. Among them, you find blood grouping devices, HPV, so all these bloodborne virus parameters, but also SARS-CoV-2 and highly virulent influenza viruses, and also, especially for transplantation transfusion, certain other viruses, but also protozoa like Plasmodium, Toxoplasma, and so on. So these are considered the highest risk, or coming with the highest risk in the case that something goes wrong with the diagnosis. And then the next lower class is class C, which means it's a high individual but a moderate public risk of if it comes to failures of the device. Examples for this are HLA typing, so tissue typing, or the sexually transmitted pathogens are among them. Pathogens in CSF and in blood. Then also pathogens that can expose a risk of death or disability to the individual that has been tested or an embryo within the mother in that case. It also comprises prenatal screening and screening of congenital disorders. Things like disease staging, all the cancer diagnostics and inherited genetic tests are class C devices. And also important to know, all self-testing and companion diagnostic devices are class C devices. So everything, if you think about point-of-care testing, really by a layperson, that is considered self-testing according to the IVDR and is automatically a class C risk device. Then I jump to the lowest risk class, which is class A. These devices show a low individual and a low public risk. This is things like general lab use, instruments, materials, buffers, culture media, also histological stains, but also DNA, RNA extraction and quantification kits. Instruments that in themselves don't generate a particular diagnostic result, like PCR cyclers, sequencers, or liquid handlers, and also specimen receptacles are considered class A. And everything else which is not mentioned here or does not match the characteristics is then grouped in class B. That's kind of a remainder classification. So there's a very nice document available, published by the European Union or by the European Commission. It's called the MDCG papers. There are many of them, and the 2020-16 paper is providing a very nice guidance of the grouping and includes also many examples. For example, those that I have listed here. So you can, following this guidance very, very nicely and easily, also classify your particular assay. So what are the implications of these risk classes? The most important implication is that it defines the scrutiny of the review of the device files. So if you see here, Class D, C, and B, so 3 higher risk classes devices, have to mandatorily have to undergo a review and certification by a so-called notified body. I will explain in a minute what that is. And only Class A devices, so general lab use, extraction devices, instruments without, as I said, generating results on its own, are so-called self-certification devices, means the manufacturer does not have to go to a third party like the notified body to ask for review and certification. So, what is this notified body? You can think about it as being an independent organization that controls IVD manufacturer on behalf of authorities in the individual EU countries. And the notified body has to perform periodic review of the quality management system of manufacturers, as well as periodic review of technical files of the actual IVD devices. So, and that is also an important point here if it comes to cost. All these Class B and C devices are grouped in so-called device groups according to certain algorithms, basically, which are all public, of course. But that means you can basically put many similar Class B, C devices with either similar intended purpose or other similarities into one device group. And then there is always just one example of this device group being reviewed by the notified body every year. So that lowers the cost compared to the Class D devices and self-testing and also near patient testing, because here it's really mandatory by the law that each and every device, Class D device or self-testing device or near patient device, has to be reviewed and approved, so certified, before the manufacturer can market that device. You can imagine that costs a lot of time, that generates a lot of cost to the manufacturer. So if you think about the certification process, you can calculate roughly 12 to 18 months from the first time you contact a notified body to getting the certificate in hand and basically starting with marketing your device. So, so now, in a nutshell, what is required to CE mark a test? I've, of course, summarized that a little bit. If you look only at the device, it must fulfill the so-called Annex 1 of the IVDR, which essentially is about the general safety and performance requirements. Show you in a bit what this is about. And you have to consider the so-called harmonized standards. They are usually mostly ISO standards, like the ISO 13485 or the one for risk management or the one for stability, for performance testing, and so on. So there is a huge bunch of ISO standards you need to consider and take into account if you want to prove to the authority, or means to the notified body, that your device is in conformance with all of them. And the other important part is that the device needs to be documented according to the Annex 2 of the IVDR, which is the describing the whole content of the so-called technical documentation. And if it comes to the manufacturer itself, these also have to fulfill certain requirements. First of all, they need to comply with quality management standard ISO 13485, which is the QM standard for medical devices in general. And as I said, also in a nutshell, they also have to fulfill the post-market surveillance and vigilance obligations that are required by the IVDR. Post-market surveillance means things like you have to periodically review the performance of your tests in the market. You have to review how much complaints you got, whether there was any feedback that could lead to improvement of the devices and those kind of things. And vigilance means if you really get complaints, then you are obliged to take measures to inform the authorities. Worst case, or I mean, normally then doing a field safety notice to your clients, to your customers. And worst case is something like withdrawing devices in the market, or say from the market.
فالبدايه بتكون حاجه بتبقى معايا انا، فلما الاقي حد من بره بيقول نفس الحاجه
Can
لا
you
عايزه
kindly
ابقى
mute yourself?
كلكم يعني.
Thank you. So what are these general safety and performance requirements? I've grouped them into three basic categories. First of all, the so-called general requirements. These are a total of 16 items. They describe things like your device has to fulfill the intended purpose that you have defined for it. It must be state of the art. It must be safe to use. You have to implement an active risk management system according to this ISO standard. You need to continuously monitor and minimize application risks, and you have to take into consideration the safety and service of the device throughout its lifetime. Then the second group is about the performance, design, and manufacturing of devices. It's a total of 60 items at maximum that need to be considered and checked basically if they are applicable, and if they are, if you can prove that your device is in conformance with them or is fulfilling them. This comprises things like the performance characteristics, chemical, biological properties, like hazardous materials, infectious materials that can be part of a device. Also infection and microbial contamination that might happen, the design, manufacturer, and interaction with the environment, and so on and so on. Some stuff just applies for instruments like the radiation, some electronic systems like software, or internal, external energy sources. And the last group is about the labeling and instruction for use. Basically, any information that you have to provide with the device. It can be directly this label and instruction for use, but also other product-related information. So this is a total of 95 items that are listed here. It is actually so much because the European Commission decided to implement a lot of these ISO requirements that used to be in separate ISO standards. They decided to put this into, pack this into the IVDR. So essentially you have now all the relevant ISO standard requirements directly in this Annex 1 here. And that's why it's so blown up if you compare to the previous regulatory framework. So, what is the technical documentation about? You see, it's quite a bit of things that need to be prepared. It starts with the definition of the intended purpose of your device, a general description of it, the composition, including recipes and everything, justification for the classification that you have chosen, and also all these information supplied with the device again, then design information, the GSPR checklist. So basically the checklist proving or showing that you fulfill everything that I have just mentioned in the slide before. The tech documentation also includes all your risk management and the risk benefit analysis and statement, where you state legally binding that the benefit of using your devices is outweighing the risks of using it. Then things like the validation of specimen type, analytical and clinical performance, product stability, the scientific validity, and then some post-market stuff like post-market surveillance and performance follow-up. So just to give you a brief idea, I have picked out here three things, like the analytical performance, clinical performance, and product stability, which I think are most relevant to you if you develop assays. Because that is something that you would need to do anyways, because you want to show that your assay has a certain performance. And it's very clearly defined what a notified body would expect here. In terms of analytical performance, it's things like the accuracy and precision of measurement, it's intra and inter-assay precision, analytical sensitivity, as well as specificity, interfering endogenous but also exogenous substances. If you think about quantitative analysis, it's the measuring range and linearity, and also limit of detection quantification that you have to determine. In terms of clinical performance, it's things like clinical performance studies or peer-reviewed scientific literature that you can provide here as part of the technical documentation, or if you have this at hand, published experience gained by routine use of this diagnostic. That usually is more applicable to legacy tests, which are in the market for many years already, and you can retrieve data from laboratories using these devices. So for the clinical performance studies, there is a rule of thumb which I want to give you, that you should test at least 100, better 150 samples for this clinical performance study. That gives you a statistically significant result and idea how your device really performs. And the last part here is the product stability. That is also very important. If you think about a manufacturer claims that the assay is stable for 2 years, then the manufacturer, of course, have to prove that this is really the case. And you can do this by accelerated stability studies, where you put things onto higher temperatures, and then you basically have an acceleration of the aging process. But at the end, you always have to show also real-time stability data. And this is also a hard requirement. This needs to be determined using 3 different batches of your final device. And then there are things depending on the mode of action, things like onboard stability, open vial stability, and you always have to also consider shipping stability. For example, if you decide your device can be shipped at ambient temperatures, then you need to take this into account because it may also influence the aging of the device. So on top of all these more device-related obligations, what are the manufacturer's obligation itself? So as I mentioned, the quality management system needs to be complying with ISO 13485, a certain quality standard, which includes things like a controlled development process, controlled manufacturing process, maintenance of devices, calibration and stuff like that. And so that's these things. But also you have to have a procedure in place that if you change things in your device after you have put it into the market, that this needs to be done in a very controlled and determined way. So that you can always trace what has been changed. Did it has any negative impact maybe on the product and stuff like that. So you is also coming with a review of your initial risk assessment that you have to have always up to date. You always also need to validate your manufacturing processes, and you need to ensure that you always can identify devices that you have manufactured within the manufacturing process, but also afterwards. And that's why traceability from the final product, even shipped to the client, to the customer, back to the very initial raw materials is of absolute paramount importance. And if it comes to this post-market surveillance and vigilance, the manufacturer has to also in place these procedures. What needs to be considered is defined in both the IVDR, but also partly in the ISO standard. And for all these assays that generate some sort of diagnostic result, they, as I said, are at least Class B, Class C, or Class D risk. That means they have to be certified by a notified body. And that means the manufacturer has to send periodic updates to his notified body about the performance of devices, failures, and complaints. That's also a must. Then there is things like this post-market clinical follow-up, where in certain cases the manufacturer has to take some effort to find out if there are changes to the clinical performance of the device. And maybe some new background information because other, let's say, mutations are becoming known. So you might have to adjust your assay to these new mutations. And then last but not least, the vigilance procedure is also very important, especially if you really face some safety issues with your instruments or devices in the market. So that's a lot of things. The question now is, what can you do as a researcher to kind of support this this whole certification requirements? I mean, if you want to bring a test to market that shall be used in routine diagnostics, then it must be certified either in the EU according to the IVDR or in the US according to the local regulation. And you have similar regulations in many different countries. So you can, for example, or I would say, look for a partner as early as possible who is experienced in IVD device manufacturing because of all the requirements that I have just listed out. And also, before you go to your partner, you should think of a business case that justifies the high certification costs. I told you you would need to count like 12 months at least from the first approach of a notified body to the certification. And for either the first device group or an individual class D, means highest risk device, you have to calculate with roughly 50,000 euros for certification. And that is only the external cost. It's not counting for any internal costs at the manufacturer. So of course, a manufacturer would be hesitant if you cannot really show that your device would earn something, because otherwise it's just a loss of money. And for a commercial business, it's viable, it's crucial to make some money. Otherwise, you will not survive. You should plan plenty of time for the device certification after you have your own stuff done. So no false expectations in terms of, I have done my stuff and in 2 months from now, this is going to be a routinely used device. That is not going to happen. And also really have a deep look into this general safety and performance requirements, this Annex 1 of the IVDR, and then try to tick off as many of the performance requirements, which I have also listed out in one of the slides, as possible, because that makes the life of the manufacturer easier, who eventually would have to compile the technical documentation file where the the performance stuff is part of it. All right, that's it from my side.